So let's first look at how cell produces an RNA copy of the gene. And the location, you guys have to know the location for this, it's the nucleus. So transcription is going to take place inside here in the nucleus. Translation takes place in the cytoplasm. Just like glycolysis, Krebs cycle, and electron transport chain, you had to know the location. Make sure you guys know the location for this as well. So transcription happens in the nucleus, translation happens in the cytoplasm. So how does DNA pair with RNA? So we learned that adenine is gonna pair with uracil because RNA doesn't have thymine. Cytosine is gonna pair with guanine. Guanine is gonna pair with cytosine, and then the thymine is still gonna pair with adenine. Only difference is that adenine doesn't pair with thymine, it pairs with uracil. So maybe make a note of that and try to remember that. So there are 3 steps to transcription: initiation, elongation, and termination. Initiation is where it starts. Elongation is when it keeps adding those base pairs. Termination is where it stops. So let me show you something before I go into detail for that. So I told you in the very beginning today that the chromosome has multiple genes. So you have this chromosome, there's one gene here, another one here, another one here, maybe another one here. So it has multiple genes. Each time transcription happens, it's gonna begin at the beginning of the gene and it's gonna go until the end of the gene. Initiation is here and then termination is here, the end. And this part is elongation when it keeps adding the base pairs or bases. So initiation, elongation, and termination. So let's zoom in on initiation. So this is the beginning of the gene. That's where it starts. So that's called initiation. And it needs an enzyme for that. And that enzyme, you need to know the name of the enzyme. It's called RNA polymerase enzyme. So it's RNA polymerase. It's going to attach at the very beginning of the gene. Don't worry about this promoter region. It is called promoter region wherever it starts, but I'm not going to ask you that. There is an enzyme called helicase. That's the one that's going to unzip this double strand. So if you guys remember, these are attached through hydrogen bonds that are weak bonds. And once this unzips, those hydrogen bonds, they break easily because they're very weak. And once that happens, this polymerase enzyme, RNA polymerase, can start copying the information. So that's elongation. So on here, you can see that it's copying the information and making an RNA strand. And when you look here, you're gonna see that for the C, it's adding a G. So these bases, they're just floating around in the nucleus. It's grabbing the complementary one and adding it. It's like a train track, adds the next one and the next one and the next one as it keeps unzipping until it reaches the end. So for C, it's gonna add a G. For another C, another G, G, it's gonna add a C. Here, A, it's gonna add a U for that. And the reason, because we learned that RNA does not have thymine, it has uracil. So it adds a U. And then for C, another G. So it's gonna continue going until it reaches the end. And the RNA polymerase keeps moving along. So helicase is opening the strand and RNA polymerase is copying the information, keeps adding the bases. And once it's done, it's gonna close back up. So let's look at termination. Now we have reached the end of the gene. So we have reached, maybe we started here and we reached the end of the gene. That's termination. So once it reaches the end, it's going to let go. So RNA polymerase separates, DNA strand separates, and RNA strand separates. DNA strand becomes double helix again. The weak bonds, they're going to start attaching again here. And now you have the copy the information from DNA and you have a RNA strand here. I want to stop here and ask if you guys have any questions. Please ask if you have questions or you need to need me to repeat something.
Is there any different enzyme in the elongation?
No, it's just the RNA polymerase. It's going to complete the elongation. So there's a question. So actually, let me go back here. I told you that it has to start from the beginning of the gene and it has to go until the end of the gene. Now, let's say that it doesn't reach the end. It just stops here. What's going to happen? Is it still going to be able to make the same protein? So in this case,
Can you repeat the question again, please?
so we're saying that it started copying the information for this gene, but it didn't reach the end. So it only has half of the information. Would it still be able to make the same protein?
No,
No.
no, man.
No, right? So it's not going to be able to make the same protein. Same thing if there's a mutation. So if somebody needs, we need a lactase enzyme to break down lactose. If their genetic information is, or their genome is missing the information to make the lactase, they're not going to be able to make that. And same thing if it doesn't go all the way, the information doesn't get read all the way, it's not going to be able to make that protein. So there's a question.
And that's how Down syndrome happens.
Yeah, sometimes there are people, they're missing that enzyme, that gene to make the enzyme. So the lactase enzyme, and that's why they're lactose intolerant. So if a gene has the wrong nucleotide sequence, what would be the consequence? What would be the end result? Kaylin said A, Finney said B. What do you guys think? Everybody needs to type an answer. Daniel said B. So answers are mixed. Some people are saying A, some people are saying B. So if a gene has the wrong nucleotide sequence, the RNA will have the wrong information. Therefore, it's not going to be able to make the protein. So this is the end result. It cannot encode the protein. So the RNA will still be able to copy the information, but it's going to be wrong information if the gene has the wrong information. So this is the end result. This is true, but this is the end result. You cannot make the proteins, and that's what you need end of the day. Okay, so we were here. We said that transcription happens, the RNA copy the information. You have single strand of RNA. Now, that RNA molecule has regions that are called exons and introns. So before the RNA, messenger RNA can leave the nucleus and go in the cytoplasm, it needs to be cleaned up. So it's just like when you're writing a paper for your class, you never want to turn in the first draft. You want to kind of look over it and edit it and then turn in the final draft. So cleaning up process takes place. And in that, these introns, they are removed and you're left with exons. So if you have a hard time remembering which ones get removed, think of introns as an intruder in the house who needs to be removed. So introns are removed and you're left with exons. These intron regions, they're still doing research on it. It doesn't have any functional, and maybe your ancestors had some function in these regions, but right now our body basically removes these parts and this is what you have. And in addition to that, there is a poly A tail and mRNA cap that's added. And this is for protection. Poly A tail is the adenine base that's just being added to one end, and on the other end, it's mRNA cap. And this is for protection. Think about shoelaces. So it has caps to prevent the fraying. So you have this cap at the end of shoelaces. So this poly A tail and mRNA cap is just like this. It's to protect the mRNA molecule. So once the introns are removed, you have only exons and you have a poly A tail on one side and you have mRNA cap on the other side. I keep calling it mRNA because this is a messenger RNA. It's taking the information from DNA and going to the cytoplasm. So once you have this, it's going to be called mature mRNA. This is your final copy. It's called mature mRNA, and it's going to leave the nucleus. So that's transcription. So here you can see the introns and then the exons. So during intron removal, introns are removed from RNA molecule, and then you have the mature mRNA molecule with the tail, poly A tail, and mRNA cap that leaves the nucleus. So we have completed transcription. Now the translation needs to take place in the cytoplasm, and that's where the information is going to go. Any questions on transcription? No questions? So there is a question for you guys. If the DNA template strand has the following sequence, this is a sequence, what would be the nucleotide sequence of complementary RNA molecule produced in transcription? Now I'm gonna have you do the transcription for this template strand and you're gonna tell me the answer.